job, I would have done it. Okay, so this is going to be the second half of the fluorescence lecture from, I don't know how many weeks ago, maybe 7, that we didn't get to. And now it seems like we should have just gotten to it and put it in earlier. So I apologize for those of you who are working on things that have to do with pigments. Maybe this will help, or hopefully help or hinder. I'm hoping it helps. So this is what we're going to try to talk about today: classifying pigments, talking about diagnostic pigments and taxonomy, how to use pigments to get at phytoplankton, how do you actually measure pigments with HPLC, what is HPLC anyway, and then estimating pigments from, say, absorption deconvolution, from single channel fluorescence, and from multi-channel fluorescence. So a bit to cover today. Okay, classifying pigments. So what's a pigment? Don't look at your screens. Don't look at the answer. What's a pigment? Something that absorbs light. That makes sense to me. So it is. It's actually a compound that absorbs light and imparts color, because of course it's absorbing some of that light away. The definition is that it is a compound that's nearly insoluble in water. And we know this, right? Because did we extract pigments in water in the lab? No, we extracted them with solvent, right? And it's interesting, and I never thought about this before, but it turns out that we can, dyes and pigments are not the same thing. And I had no idea about that. So it turns out that dyes are soluble in water and they actually bind onto other things like fabric. And that's how they give color. So that's really different from a pigment, which just is not insoluble in itself, it absorbs light. So, okay. Biologically, we think about pigments as they relate to phytoplankton pigments. And we can separate them into those that absorb light for photosynthesis and those that absorb light to protect phytoplankton from light, from too much light. So photoprotective and photosynthetic. Okay. So I've divided this into 3 different major types of pigments, and the first one are the chlorophylls. And so the chlorophylls, we have chlorophyll A, that's the dominant pigment. The reason why we use it as our indicator of biomass and phytoplankton and photosynthesis and everything else is because it's dominant and ubiquitous. All phytoplankton have chlorophyll A, and only phytoplankton have chlorophyll A. And so it can be that really unique proxy. I mean, of course, seaweeds have it too, but we tend to not measure those. We hope to not measure those in our chlorometers, our dirty chlorometers. So here's that basic structure of the ring, and with all of these different residuals, and you can see that depending upon what that residual is, we have chlorophyll A and B and C1 and C2 and C3. Here are the C pigments. I'm double handing here. Oh, I'm supposed to be using this. Okay. So chlorophylls B and C, phytoplankton generally don't have both. Chlorophytes have chlorophyll B, and non-chlorophytes have chlorophyll C, and they may have one of chlorophyll C1, C2, or C3. And what those pigments do, you can see by these spectra, the green here is chlorophyll A. This is from Ricot, so we have the whole issues with exactly the ratio of the peaks. But then the chlorophylls B and chlorophyll C are greener in their absorption, and they're also, more towards the orange. They're narrowing the absorption window of the visible. So they're allowing phytoplankton to absorb light more towards the green end of the spectrum, where light tends to be in the ocean, right? Then we also have the divinyl chlorophylls A and B, and those are found in Prochlorococcus. And because they're only found in Prochlorococcus, we call it a diagnostic pigment. If you have divinyl chlorophyll B, you have Prochlorococcus. And what you can see from those is they're just slightly different from the chlorophyll A and slightly different from the chlorophyll B. Okay, then we have carotenoids, and those also are filling in the spectrum, but in the blue-green part of the band. And so I've divided them into the groups that I look at when I'm thinking about who's in the water. So we have fucoxanthin, 19-bute, 19-hex, and carotenoid or pyridinin. And you can see they're really similar in their chemical structure, right? There's long chains of carbon with double bonds that's responsible for the resonance that leads to absorption. And if you look at the spectra of these, here's fucoxanthin, diatoms, we think, right? Here's pyridinin. Oh, dinoflagellates. Look how different their spectra are. It's amazing. We should easily be able to resolve that with remote sensing. Not. But that's okay, because now we have 19-bute and 19-hex. Okay, so they have, it's 2 peaks or less or more resolved, but honestly, when we are trying to distinguish between diatoms and dinoflagellates, we are looking at the differences in the ratios from here to here. Yay, hyperspectral. So Jeremy's going to talk about this afternoon, like how much can you wring out of a spectrum? And I will tell you that sometimes you can. Sometimes if you don't have a lot of CDOM or you don't have a lot of competing things, you can see differences in this sort of blue to green ratio, differences in the pigments, but it's really difficult to do that. It's a challenge, but we're blasting forth with that. Okay. The next set are the Diatoxin, Zeaxanthin, and Alaxanthan, which also tend to show up in specific pigment groups. And these have a really distinct absorption compared to the fucoxanthin, pyridinin 19s. Right? You can see that they are very, very different. And so looking at phytoplankton that maybe have fucoxanthin, but also maybe have some of these other pigments might help you that might be what is helping you distinguish between diatoms and dinoflagellates. If they have different pigments of this group, okay, the zeaxanthin, alaxanthin. And then the last group are the carotenoids, or the carotene. So this is alpha carotene and beta carotene. And sadly, they're really similar to this diatoxanthin, zeaxanthin, alaxanthin. But this is what we're dealing with. Okay? And again, these are in extract, but you get the point. Can quickly identify the differences in this structure here, and you can see why we have different peaks, but okay. Then we have Psychobilly pigments, and these are associated with cyanobacteria and certain of the rhodophytes or green phytoplankton or red phytoplankton. We have phycoerythrin, phycocyanin, and allophycocyanin. And these are very different, and they tend to not be soluble in methanol or acetone. These are water soluble. I wouldn't call them a dye, I would still call them a pigment. But anyway, this is what their absorption spectrum look like. So tell me why would an organism have these pigments? Coastal waters, right? Look at this. Like whoever has phycoerythrin is going to rock it in these waters, right? Because they're absorbing really efficiently at the 550. And what about these? Where do you think those evolved? Where?
Red blue.
Yeah, red water. So like places where there's so much CDOM that really the only light that's left is out in here. And in fact, these did tend to taxonomically evolve from species of bacteria that were living in basically red waters. Okay. Yeah.
What are the absorption coefficients of these pigments compared to substantially lower?
The specific absorption, or it depends on how much pigment you have.
So if you had a sample or average amount of phytoplankton that have mostly chlorophyll A and then also have these pigments, would it be a full beta?
That's like a Seneca cox out in the open ocean, and you get these big giant peaks. And in fact, in exports in the North Pacific, when I size fractioned the water and I looked at the less than 5 micron fraction, that was living down around 140 odd meters. They looked like this. They had a big giant peak here. When you looked at the filter, it was pink. Yeah. So these can be major pigments. If they are a major component of the phytoplankton community, it shows up in your absorption spectra. And when you extract with methanol on your filter pad, it doesn't extract. So you have to extract it with a phosphate buffer or something else to get rid of that. Yeah. So what this paper was showing was that you find Chlorococcus or Synechococcus or I don't even know what species this is going to be. And it shows that the pigments that they tend to have in terms of Psychobilly are matching the spectral nature of the water that they live in. So here, Chlorococcus lives in the open ocean where the irradiance is maximal at 490. And they have this really nice pigments, divinyls, that allow them to get in there and absorb that. This one is going to be in the very green waters or down deep where there's only green water. And then this one is more in the reddish waters. Okay, so yay, we have all these pigments. We know that certain phytoplankton have different types, different individual pigments, and can use those to tell us something about taxonomy. That's really the basis of PACE, which is we have hyperspectral. That should give us pigments, and that should give us taxa, right? That's how it was sold. So here we go. This is from a paper by Sasha Kramer, where she's outlined the major pigments and the groups. These are the sort of red lineage, such as diatoms, dinoflagellates, chrysophytes, haptophytes, cryptophytes. The green line, presentophytes, euglenoids, and chlorophytes, and then the cyanobacteria. And then she went through the literature and figured out which pigments are unique to a specific group. That's the dark color. Which pigments are always found, which are sometimes found, which are never found, or which are not present at all. And so this is what you're dealing with. So, so some pigments or combination of pigments are what allow us to try to get at taxonomy. And so that's great, but for every example, there are exceptions. So the first one is, yay, divinyl chlorophyll A and B is Prochlorococcus, and it's only found in Prochlorococcus. So if you have that, you know what you have in the water. So check for that, yay. Okay, let's look at this, chlorophyll B, right? We've talked a lot about chlorophyll B. And what we see is that chlorophyll B, except for certain, except for certain like dinoflagellates, some have some chlorophyll B, but usually it's all of the green lineage. And so you know that chlorophyll B is, if you have chlorophyll B, you know you've got some of these green algae. But which ones? Well, we start to look at these pigments now. And so if you had Alaxanthan, you would know that that chlorophyll B is associated with chlorophytes. If you had Diatoxin, you would know that your chlorophyll B was associated with euglenoids. If you had none of these, you would know that your chlorophyll B is associated with a presentophyte. So that's the type of sort of thinking you have to do when you're trying to use pigments. It's like, well, I have this, but I don't have this. Yeah.
What is the difference between those two types of chlorophyll A and
The chlorophyll A, chlorophyll B. So monovinyl chlorophyll A is chlorophyll A that you're used to measuring in the lab, and the divinyl is a slightly different molecular structure that is only found in Chlorococcus. So it's very rare, except for you have Chlorococcus, and it dominates numerically in the ocean. Yeah, tiny but mighty. Yeah.
Is there a feel for how much of that is needed to?
So dinoflagellates are a weird bunch, right? They are phototrophic and heterotrophic and both. They have symbionts. So there's lots of dinoflagellates are just very, very broad. They're very difficult to identify because of their broad taxonomic and evolutionary characteristics. So one of the things about dinoflagellates that we can look at is the presence of carotenoid. And this says often present, I would say, for most of the groups that you will encounter in the ocean, they will have carotenoid. But you will always find one that doesn't, right? But if you find carotenoid, you know that you have dinoflagellates. So it's necessary but not sufficient. Yeah, or sufficient but not necessary. No, that's right. So that's a good one to look for. But of course, we know optically it looks really similar to fucosanthin. So that's a toughie, but at least when you have that pigment, you know. Fucosanthin, people say fucosanthin, we've got diatoms. Yay, that's a perfect indicator. And you will read a ton of papers that tell you that. And it's not the case because all of these groups have fucosanthin, right? And so then you have to get back to like, you know, whoops, oh, okay, if I have my 19 hex and 19 bute, yeah, I've got dinoflagellates, but if I only have 19 bute, it's a pelagophyte. But haptophytes and dinoflagellates is really similar, except that this one has more diagnostic. So you see how it happens. You find yourself down these rabbit holes. Like, if I have this, I must have this. Oh, wait, but I have this. And then, of course, you're doing this in a mixed phytoplankton community. So it's complicated. So what's a person to do? Okay. Well, we use a statistical approach. So how many people have heard of Chemtax? A couple people have heard of Chemtax. So Chemtax was first published in this paper by Mackey in '96. And basically what it is they looked at characteristic ratios of pigments across taxa. And they said, well, if you had a haptophyte, you would expect it to have this chlorophyll to fucosanthin to diadenozanthin ratio. Whereas if you had a diatom, you would expect a different chlorophyll to fucosanthin to diadenozanthin ratio based upon cultures. And so if I had measured chlorophyll, fucosanthin, and diadenozanthin in the water, I could say, well, it's probably 80% diatoms and 20% haptophytes, right? So in doing that, you could take a suite of measured pigment composition and pigment concentration from an environment. And you could partition the pigments based upon the ratios that were specific to taxa and come up with estimates of taxa. So here's an example from people who weren't involved in the development, but who used it, and I think did it well, where they went out to a series of 6 stations in one part of their region, and then another 6 stations in another part of their region. They measured how much chlorophyll A was at each station, how much fucosanthin was at each station, 19 hex and 19 bute, etc. So they have 12 stations and they have the full suite of which pigment was there and how much was there. Okay. Then this step 2 is that you know something about your ecosystem because their diatoms might not be like your diatoms. Their diatoms being the original paper of Chemtax. So if you know something about your diatoms and you know something about their pigment ratios, you tune those ratios to your region. And I will just say that most people who use Chemtax do not do this. And so their results are biased towards the community that was used to develop the original Chemtax ratios. And that is, yeah, go ahead.
Do you know
Yeah.
what happened?
Yes. Because these guys are Australians and they did all their work out of the Australian waters heading south.
So very representative.
Yeah, the Southern Ocean is known to be really different from everywhere else in the world. So if I ran Chemtax on our pigments, I would report that as Southern Ocean equivalent, you know, diatom community, dinoflagellate community, etc. So it's really important to understand the tuning that has to happen and why it's important. Okay.
Are there like tuning parameters for different bodies?
You can find papers where people have done it. And if they haven't done it for your region, you know what you have to do. You have to get your pigment data. You have to get your IFCB data. You have to do some biovolume estimates. You have to do some pigment per cell estimates and be able to tune up those ratios. Or say that we never have haptophytes, ever, ever, ever have haptophytes. So if we see those pigments, they're in something else.
When a sample sent, like Hormone Lab, a lot of samples are sent to their HPLC. How do they know how to compare.
They're not tuning. They're giving you a pigment, and I will show you how they do that. And that's the pigment that they give you is the pigment that they determined with HPLC. It's how you use the ratios of the pigments to say something about taxonomy. That's the tuning. Once you get the data, right? So you get the data, you tune for your region, and then you figure out what's there. Yes.
So when you try to go for
Yes.
I don't know, because I've never seen a paper that actually tried to see whether it varies based on consistency. How stable it is. It's a method that's out there, but you're not really thinking about it. And therefore, the assignment of the survey is that.
There's space for making this better, this approach better. It's just a statistical approach. It's just a minimization like anything else. But you just have to know what's going into it. It's like putting in eigenvectors, but putting in Prochlorococcus eigenvector for the Gulf of Maine. We don't have Prochlorococcus here, so you wouldn't put that into your version. Similarly, you know, what you want to do is make sure that you're tuning this to be appropriate for your region. Garbage is garbage out. All right, so then what you get, once you've collected all your HPLC data in the lab, you've done a good job, and then you tuned Chemtax to the fact that you're in a subpolar region or a subtropical region, you get out estimates of the community based on pigments. And so here's their stations, A1 through 6, B1 through 6, and it's partitioned. And you see things like, oh, at A1, A2, and B1, B2, lots of diatoms. Well, we could go back really quickly and look and figure out why that is. Well, they had a lot of fucoxanthin. And they didn't have fucoxanthin at the other stations, right? So part of that fucoxanthin went into diatoms. It probably also went into other things like haptophytes and presenophytes, not presenophytes. Yeah, haptophytes. And so you can begin to look at these results and figure out why they ended up with Prochlorococcus. Well, they probably had divinyl chlorophyll in those stations, right? But that's what Chemtax gets you, is an estimate of pigment-based taxonomy. And you can run it in Excel. It's worth running and varying and seeing how sensitive your model is to this type of result. Another is something called using diagnostic pigments to get at the size of the community, right? And so in the Mediterranean Sea, pigments, when they measure the HPLC pigments and they look under a microscope, they tend to find that there are pigments that are associated with specific groups of phytoplankton. And those groups tend to have specific cell sizes. And so Vidusi in their 2001 paper said, well, when we see zeaxanthin and chlorophyll B, that is always associated with picoplankton in our stations. And when we see Alaxanthan and 19-hex and 19-bute, those are nanoplankton sized species. And when we see fucoxanthin and paradena, that's when we have these really big diatoms and dinoflagellates, and they're microplankton size. So for their environments, and I've looked at these data, I've collected data like these in the Mediterranean, and it's very robust about looking at how these pigments are packaged in these different size cells. And so not only you're not getting taxonomy, but you're saying, I'm going to be dominated by nanoplankton because I have a ton of this at this particular time of year. And so they use pigments to get at this pico, nano, micro size class for the Mediterranean. And it's a really cool paper, you should read it. So because they're able to get at size classes, and then they can plot the size classes, you know, across the Mediterranean and look at seasonality. And in fact, when you get your pigments analyzed by NASA, they do this analysis for you and give you estimates of pico, nano, and micro. And that's cool, right? Uh-huh. Yeah. You see a problem with it though, maybe? Would we want to use it? Why? The Mediterranean is also a really unusual body of water. It's like really unusual phytoplankton. But people will just use this in the same way that they use contacts, just kind of blindly, right? And they've come out and they've tuned these, they have updates, different authors updating with like different coefficients that are in there. But it's, tuned for an environment. And so you have to be really careful.
And then you use it to validate the side algorithm.
Right, yes. So, okay. So the question is, does it work at other locations? And so Ali Chase published a paper in 2020 where she tested 3 of these sort of tuned diagnostic pigment algorithms. One of them was the Medusi, which was updated by Julia Wheats in 2006, the Harada algorithm, and the Devrid. And they're all basically a similar approach to try to get at size. So she collected a data set from the North Atlantic for 4 different seasons. And this is the range of stations and color-coded by chlorophyll concentrations. You see there's a big range of chlorophyll concentration. Here's where it's plotted in temperature, salinity, space. You can see it. We're looking at a variety of different zones, sort of more coastal zones, more offshore zones. And so the first thing she did was she partitioned the biovolume into size bins. And the pico, nano, and micro. And she looked at the fraction of biovolume that you would predict from this diagnostic pigment algorithm and how you would partition them by looking at flow cytometric data, okay? And so what she found was that for the picoplankton size cells, some of them really underestimated the biovolume of picos and some really overestimated based upon how they are using pigments to tell them something about size, okay? In the nanoplankton, well, I don't know, some of these points are close to mine, but look at this. So they're really underestimating the nanoplankton in general. And that's a problem because most of the ocean is nanoplankton, right? And then when you get to the microplankton, they're really overestimating the microplankton. And why do you think that is? Who is typically called microplankton? It's a big who? Which taxa do we look at? Diatoxin, Dinoflagellates, right? And so what's happening is as soon as they see Zeaxanthin, they're like, microplankton. Okay, then they partition the chlorophyll. And so this is again the chlorophyll based on cytometry versus the chlorophyll estimated from the pigments and partitioned into various sizes. And you can see that when you're in low chlorophyll waters, 0.01 to 0.1, almost every group is overestimated, right, in terms of the total chlorophyll. And when you get to this mid-range of chlorophyll concentrations, they're overestimating this should be underestimated. Overestimated the microplankton and underestimating some picoplankton, definitely underestimating some nanoplankton. And that just gets worse when you get to these larger sizes. They really underestimated nanoplankton and somewhat are putting it into microplankton. And so the problem is the models are going wrong because they're attributing size and pigmentation to the same drivers. And the one thing that I would just point out to you, when they actually looked at the size distribution, the histogram of size for diatoms measured with IFCB, 20 microns is the division between nano and micro, right? There's a lot of nanoplankton that are diatoms. And so that's where some of these algorithms are going wrong. And this is, relatively new thinking. And so this becomes really important when we're thinking about the role of diatoms and sinking in carbon. We have to think about diatoms as not just being the big, beautiful things that we see in the microscope that we are so attracted to. Okay. Another approach is the hierarchical cluster analysis. And this was done by Dylan Catlett as part of his PhD thesis, where basically he took all this HPLC pigment data and then did a cluster analysis on it. And what he found for this data set, which was from the Plumes and Blooms program off the coast of California. It's an upwelling zone. They collect data, I don't know, every month or so for years. So it's a very large spatial and seasonal data set for a small region off of California. It's a beautiful data set. And what he found was that the pigments tended to cluster into these 3 different clusters. And one of the clusters was chlorophyll C and carotenoids, or carotenes and fucoxanthin and diagnostic. And they're like, well, that's probably diatoms, right? And we look under the scope, you see that. And you get this little peak of paradigmin, which is not exactly clustered with these, a little bit further distance, but they tend to be associated with each other. Then you have all these pigments that cluster together, which are characteristic of nanoplankton, here are haptophytes and picoplankton. But you can see that size-wise, in the way that we think about phytoplankton in sort of this 3 different sizes, they are seeing that clustering in their data set, which is spatially and temporally resolved. And so then you could look and say, well, when does this cluster show up? Oh, it's showing up during the upwelling events, because and that makes sense. Diatoms tend to show up, right? Yeah.
Input to this spectrum of
the composition and concentration. So sample 1, here's your concentration of chlorophyll, fucoxanthin, paradigmin, 19 type, 19 and so you put those in as individual resolution, each data set. So for each location of time. So every point of space of time, they're not resolving the spatial temporal relationships. They're just resolving what co-varies with each other. So then Sasha took a global data set and did the same analysis. And what she found when you go from, say, a very small, well-defined system to a larger system, is that she also found clustering that made sense. Diatoms and dinoflagellates tend to cluster. They cluster differently from haptophytes, from green algae, and from cyanobacteria. And what's interesting is that she then took this and broke it into regions and then found, of course, for each region, those clusters are different, which tells us that the ecosystems are different and that the composition of the phytoplankton are different. And so it's an interesting analysis. She actually did it for the data set we have here. And so if you're interested to see what she found, you should go and look at that paper. And I would say that one of my honor students replicated it with a larger data set from here and found exactly the same results that she did with an expanded data set. So it feels relatively robust in terms of what we see and what we see in the ISCB. But again, it's just looking for covariability between pigments. Okay. So those are some tools that you can use for pigments. How do we get to pigments to begin with? I mean, how many people have heard of HPLC? Hopefully everybody by now. Oh. Okay. So high performance liquid chromatography. It used to be called high pressure liquid chromatography, but now it's performance because it's not just about pressure anymore. It's about temperature and optimization of systems. So the basic operation is you separate a chemical mixture onto a column. It allows you to separate individual compounds from a mixture. And so our chemical mixture is going to be our extract that we extracted either in acetone or methanol or whatever we choose. And that is carried by this liquid mobile phase, which is basically a solvent gradient. We use methanol and acetone in various concentrations, 90% or 80%. And it's carried through this solid stationary phase, which in our case for pigments is a silica resin. And then because of the different polarity of the molecules, they will separate out in time. And so some will make it to the end before others, yes.
Is this the same basis by which you use
I don't know. Who does gel separation? Is it on column? It's similar in the way that you're applying something to the mixture that causes different compounds to separate spatially on a matrix. So I just don't know that. So the answer is no. And then what's great is because it does that, you can actually, I don't know why they use a burette here, it's very funny. But you can actually, once you know how each of these compounds is separating, you can collect it and then run this purified compound and identify it, right? And because each of our pigments has a really characteristic absorption spectrum, you can run the absorption spectrum on these, what's extracted, and then identify the pigment. And so you can associate who's coming out first with pigment A.
You're putting that in the spectrum.
Yeah, that's how they used to do it. Now what they do is they actually have a spectrometer on the HPLC, which is beautiful. So you get this detector, you get a display, what's coming out. So for us, we know that absorption is proportional to concentration. So if it's measuring the absorption of the material that's coming out, the higher the absorption, the higher the concentration, right? So you can get concentration of pigments. And if you're measuring the spectrum, then you can identify it. And so that's the basis of how it works now. Oh, this is just why it's called performance. Okay. So I have one of these. I'm just learning how to use it. So we use two different solvents. We're following exactly what NASA does. So this is NASA's protocol. You have two different solvent carriers, this methanol and tetrabutyl ammonium acetate and 100% methanol. And they're sort of alternating going through the column and carrying pigments out, basically separating them by their chemical structures. Again, okay. So this is when you start, this is how much time you start varying the different concentrations of these two solvents. Then the sample gets injected. So we have all of our samples sitting here. Ours are not these different colors, but you know, they get injected into the system. They go through the separatory column, and then they're going to go through the absorption. So it takes about 36 minutes per sample. So we set ours up to run 24 hours. The detection occurs in here. That's where it's measuring the absorption of the signal of each one of these compounds as they come out of the column. And you get something that looks like this. So this is a chromatogram for chlorophyll. And you're basically putting the sample in and you're waiting a really long time for nothing to come out. And then suddenly you get a peak as that little plug of chlorophyll molecules goes past the detector. And if you integrate under that peak, you can relate that to the concentration. Bless you. And then at the same time, it measures an absorption spectrum. And we're like, hey, 430, hey, 664. That's what chlorophyll absorption looks like in this particular solvent, and there are my harmonics, etc. So we can identify it. This is from a sample that my students collected in Hartswell 2 years ago now. And you can see that there's a variety of different compounds coming out at different times. And those are each associated with an absorption spectrum. And so we can go back and identify like who was this and who was this. And so from that, that's how you get the pigment concentration for each sample. Yeah. Yeah. Well, because they come out at different times. They come out at different times. And so one of the things about this performance is that you are optimizing the gradients of the solvents going through the column so that you can separate molecules that look really similar, but have slightly different chemical bonding structures and slightly different polarity. I know I'm thinking that it's polarity on this side, but they can separate that even when they're so similar. But what you're trying to do is if you try to do it too fast, all this stuff squishes together. So it's drawing it out. It's making it last a long time so that you can physically, they're coming in together and then as they go through the column, they separate. You can get distinct peaks. So, you know, NASA's done a really great job in their lab of being able to separate these. Yeah.
So some of those peaks visually by eye look a little close together.
Yep.
Can that be a problem?
It can, and so this is just one of the wavelengths. This particular instrument detects at 2 different wavelengths. So as the material is going by the detector, there's actually 2 light sources, and so you can measure the peak response at, say, 440 and the peak response at 660. And so some of your pigments might have a signal in the blue, but not in the red. And so those two peaks may separate based upon the wavelengths that you choose. And so you buy an instrument that allows you to select two different excitations so that you can distinguish between things that are close together. It's quite clever. The new systems are really amazing. And this is my instrument is like a teaching HPLC, so it was reasonably priced, and yet we were able to replicate, you know, at least the method-wise what we're getting from grown-ups. Yeah. Oh, yeah, absolutely.
There's no, this program we call it.
Yeah, you have two guys.
Figure out what the area.
But you have to run the standards with every set of samples because it will vary every time you turn the instrument on. It's, I mean, the order stays the same, but you can never say that exactly at 12 and a half minutes, exactly that's when chlorophyll came out. Because sometimes it's 13, right? And so everything shifts in. So you have to run standards, and standards are vastly expensive. So making your own standards, actually collecting what comes out, with make your own standards is really useful.
What is the reason price?
A couple of $100 per pigment, and they don't peak. Oh, for the teaching, this one was like on the order of 25,000.
What's the quote?
I think the grown-up versions are like 100,000, depending on all the bells and whistles that you have. You know, like everything's 100 grand, right? 50 to 100 grand. Okay, so absorption deconvolution. So this is comparable to what we heard this morning. And Patrick, isn't what you did was the absorption deconvolution? Yeah. Okay, so basically what it says is if I know the absorption spectrum of my pigment, and I know what the concentration of my pigment is, I should be able to reconstruct what phytoplankton are, right? So that's a forward model. And one of the earliest examples of this is from Bob Bittigary in a paper that he presented at an ocean optics meeting, but never published, sadly, in the literature, which is unfortunate because at the time it was really, you know, state of the art. So he took these pigments, or the spectra multiplied it by the pigments, and you notice that he combined all his carotenoids together. And he combined his photoprotective carotenoids together. So he didn't do individual pigments except for the chlorophylls. And then he reconstructed an absorption spectrum. So does yours look as good as that? Actually, yours look better than that, right? And one of the reasons is that he did a pretty good job looking at the shifting the peaks over from being in a solvent to being in a cell, but he didn't package them. And that's why his is really peaked versus the measured spectrum, which is very smooth, right? So here's some examples of people who have done that. Oh, and this was, if you read Anya Burko's paper where she did these, she found for many of her samples that she couldn't reconstruct the measured spectrum. And so they calculated what this sort of missing stuff was, and they have a whole long explanation of what they think that is. And so, you know, factor 2 between friends, right? It happens. And what's there? Looking at the residual can be really illustrative. Okay, then there's the inverse model, which is I've measured this spectrum and what's in it. And so here's an example from a PhD student, I'm just blanking on his name, Jan Crane. And he did work in lakes, and he measured the absorption spectra of extracts and then used the absorption spectra of individual pigments in those same extracts to try to reconstruct what was there. So I know that Sebastian, you guys measure the absorption by extracts. And so now if you're minimizing to the extract, you don't have to account so much for packaging and peak shift, right? You're comparing apples with apples. So I actually used this approach for my students, and I wrote him an email and just asked him for his spectra. It turns out he was just finishing his PhD. He was so excited someone was going to use it. He gave me all of his data, he gave me all of his code. He's just an awesome guy. So, you know, if this is an approach you want to use, contact him and he'll like come over and help you. He's really great. And you can see that between the observed and the reconstructed, you do a really good job when you're comparing solvent pigments with solvent extracts. Then there is looking at pigments from the perspective of the Gaussians, and this is what Emmanuel was talking about. And so you recognize that you can reconstruct an absorption spectrum by summing up various combinations of Gaussian features. And so the first step is to figure out which Gaussian spectra you need and how much of each one that when you sum up will give you a phytoplankton curve. And you have to add a little bit of the non-algal particle part of the phytoplankton, right? Because they do have cellular material. And then the next step is to say, well, which of the, we have a lot of this peak. Who is that peak? And so then you have to do this relationship between the peaks and the pigments. So you have HPLC pigments, you have Gaussians, and you try to relate them so that you can say, well, I have an awful lot of this peak and that peak is associated with this.
That was adding to the question, but if you're taking spectra, taking extracts from that spectra to recreate the spectra, what exactly are you doing?
So these were measured, these are independent spectra. They were not part of that. Yeah, they're from a library that he created from standards that he bought. Yeah. It's basically the eye detector from an independent set. Yeah. Yeah.
But I don't fully understand the benefit of the Gaussian curve first having the index. Is it if you don't have the pigment indexes, or
It can identify for you places in the measured absorption spectrum that you have not adequately parameterized with, say, your average phytoplankton item vector or your diatoxin item vector. And so if you're putting in, if you say like, oh, all I have out here are diatoms and dinoflagellates, and you do your inversion and you end up with this place where it doesn't quite fit, a Gaussian can help you identify the peak and the width of an absorption that you're missing. Right? And so then you can say, well, what is that pigment? And it could be gyroxanthin diester. And then you're like, oh my gosh, I have Carotenoid brevis out here. Right? Which would really be bad if we got that one too. Although the domain seems to love all of its toxic dinoflagellates.
so you basically need a lookup table with these Gaussians.
Yeah. Exactly. Okay. All right. So I just wanted to follow up with how we estimate pigments from a single channel fluorometer. And this was just a fluorometer that we use, but we didn't get to, well, we talked a lot about like, I have a fluorometer, it's exciting at 460 or 470 and measuring fluorescence at 695. And you guys have done that, you've done it in the lab. We also had you look at the difference in the calibration, depending upon who you calibrated your fluorometer with, and you found differences. And you found differences if they were in ambient light or kept dark, right? And so you also may have observed a somewhat of a fluorescence maximum at depth because the fluorescence of the surface might have been inhibited. We see that at the morning. And so that's not supported by the chlorophyll value. And so we talked about non-photochemical quenching. And so how do you deal with this? And so I was going to show you, well, here it is. Here's the chlorophyll profile. This is, everyone uses this. Paul Falkowski, John Raven. So they measured fluorescence in the morning, this profile, and then they went back and they measured it at noon. They probably stayed out there on the boat. I'm guessing, I didn't ask them. They also collected water samples and did extracted chlorophyll. They don't have error bars, any uncertainty. I have no idea how good they are or what they do. I'm assuming pretty good. But what you see, of course, is that the chlorophyll, extracted chlorophyll did not change, but the fluorescence did. It got it decreased at noon, and that is non-photochemical quenching. So they have a nice description about that in this book. You can also see it on a profiling float. Here's an example from Zing's paper where the profiling float was there at 8 p.m. Sorry, I can't do math up here. And versus doing it at 6 in the morning. And I don't know if those were actual, they're probably actual times, not GMT. And so you just see that there's, in the same location, there's an inhibition of fluorescence on the profile and float. Here's an example from some glider work that Mary Jane and her student Brandon Sackman did, where their gliders did this repeat path around Washington. And you can see that the fluorescence sort of always goes down. Midnight is the dot, is the red line, so noon is in between. And you can see that the surface fluorescence is quenched, at least this really interesting spatial pattern that if you didn't know was quenched, you would interpret as low chlorophyll. You say, chlorophyll's so patchy, it's crazy, right? But that's, and you would see it over space, not time, right? Here's an example from a wire walker from Melissa's work where she measured PAR and measured fluorescence, and you can see exactly what fluorescence is quenching. So I could go on for thousands of examples. Here's the last one. So this is from the morning. Here's PAR in blue, and you can see that at noon, the chlorophyll gets quenched. And it's titled here. I don't know. It could be real. It was real. You can exactly see that when you have less light, you have less quenching. You can see when it recovers. The interesting thing is that, oh, this wasn't LOGO, actually, this was offshore because I had an absorption meter out there. And what you see from the absorption meter is that there was no change essentially in chlorophyll over the day from the line height. So that's how we established that it was quenching. And if we looked at the ratio, which I normalized to 1, if we looked at the ratio of fluorescence, chlorophyll by fluorescence versus chlorophyll by line height, you can see that as the sunlight increases, we get quenching, right? And for those of you who are used to looking at P versus I curves, you can think of this as when light is really low, your photosynthesis increases linearly with the radiance. And you can see that the ratio of fluorescence to absolute chlorophyll stays constant. But as soon as you start to quench in high light, that's when you're also quenching your fluorescence, right? So you can fit curves to this and use that as a correction for non-photochemical quenching. But the other thing that people do if you have a mooring is you're like, well, I'm just going to draw a line. I'm just going to extrapolate from midnight to midnight. That seems pretty easy. And I can estimate what my chlorophyll is at noon. And then I can use that to validate my satellite estimated chlorophyll, which is what my student was going to do. And nothing, it wasn't working. The values that he extrapolated for noon were not anywhere near what he had anticipated by the version of chlorophyll algorithm. So we kind of, so that's one thing. So I'm going to show you what ends up happening with this. But another way, if you have a profile, is just to say, well, it's probably uniform in the mixed layer. And so I might have quenching, but I'm just going to say whatever my, mixed layer depth is, I'm just going to extrapolate to the surface. And that's what they do routinely on the profile inputs. You don't have other information. Another thing that people do, and this is from the glider data, is they look at the fluorescence to backscattering ratio. And what is fluorescence to backscattering ratio? What is that telling you? If you're thinking in terms of proxies. Yeah, so it's basically what fraction of the total particles are phytochemical. And so essentially, if my backscattering doesn't change, then probably my phytochemical is not changing either. And so you can use the backscattering, the fluorescence backscattering at night, it's basically uniform. The next night is basically uniform. And in the day, it went down because fluorescence was quenched. So you basically say, well, it's uniform here. I'm going to extrapolate to the surface. So if you have a backscattering sensor, that's an excellent assumption to make, as long as you're within a mix. All right, so back to my student who was trying to figure this problem out. And one of the things that we noticed was, here's chlorophyll fluorescence, here's light, this is like over a month, here's some currents out at LOGO, really strong currents. What we noticed was, of course, at noon time, this is the dark here is nighttime, at noon we saw quenching. That made sense. But then we also saw quenching at midnight. What? Quenching from the moon. There was nothing to explain it. And we looked at a lot of physiology papers. We couldn't find anything. And it turns out, you guys know what it is, it's tides, right? Tides are carrying different water masses past the mooring. So what he did first was he took all of his observations and he figured out which ones were quenched based upon irradiance. And those are the gray dots, right? There's a lot of quenching out here. Then he identified which of those hourly observations were associated with the high tide, highest, like maximum high before the turn, and which were associated with the low tide. And he color coded those. So high tide is blue, low tide is red. And then he identified which of the high tide observations were unquenched and which of the low tide observations were unquenched, because of course, the diel cycle is 24 hours, the tide cycle is 24 hours and 48 minutes. So there's a bias that happens. And then you could just connect those dots essentially as a first run. And what you see is that there are times when the populations between high and low tide are really similar, and times when the populations are really different. You're looking at very, very different communities, right? Oh gosh, not this. Oh, please let me get out of here. Okay, all right, that's it. Okay, so then what he did is he fit a tidal sinusoid to the high and low tide events that were unquenched. And then he looked at all of the unquenched data, and it looks like that model fits the data pretty well. Sometimes it doesn't. That's telling you something about ecosystem growth. And what it allowed him to do was to get an hourly observation of chlorophyll fluorescence, even at night, even during the day, based upon the assumption that over a 24-hour period, you are mixing between two end members of upstream communities and downstream communities. And I've actually applied this model to mooring data collected all over the Sound and also at Rousseau. And it helps you, there's enough tidal variability in the populations that are moving past the mooring that you're able to actually reconstruct noontime estimates of chlorophyll from fluorescence in the absence of quenching. So it has assumptions of mixing between this high and low communities, but it actually does a really great job out there. If you don't do that, here's your validation between fluorescence and extracted chlorophyll. If you do it, he wasn't a very good chlorophyll estimator, but you know, with his uncertainty, which are encapsulated within his dots, it's a better fit. So, okay. The last thing, what time is it? I'm gonna do really quickly through multi-channel fluorescence. It is not a new idea. Using different excitation wavelengths. This is the typical one for 70. You could excite in the blue, you could excite in the green, and you're exciting different pigments, and they're passing their energy on to chlorophyll and fluorescing. So it's probing pigments. Okay. So here's a quiz. I've got two phytoplankton, population 1 and population 2. And I have a fluorometer that excites at 440. And what is the relative fluorescence I'm going to get from these two? Which one's going to give me the most fluorescence? Don't overthink. Yes. More absorption, more fluorescence, right? And in fact, it's almost a factor of 2. This might be a dilution series, right? So the magnitude of fluorescence gives you an estimate of the magnitude of absorption. What about one phytoplankton that I'm going to excite with 3 different wavelengths? And I'm going to measure the fluorescence. I'm going to excite with 440, measure fluorescence, excite with 470, measure fluorescence, excite at 532 and measure fluorescence. Based on this absorption spectrum, which wavelength is going to give me the most fluorescence?
They're all in what they do vibrational states, but that is same
Well, the per photon, yes. But how many photons are being absorbed in the blue, green, right? So in fact, you're going to get the most fluorescence when you excite here and the least fluorescence when you excite here. because of the absorption. Yes.
Does it depend on whether, how much of this absorption goes east versus?
Somewhat, but that is a small source of variability compared to the difference in the absorption coefficient. Small. That's an efficiency. Yeah. And there could be photoprotective pigments in here we haven't touched, right? But you can say that you will get more here than here than here, right? And in fact, that you're probably going to get twice as much here as here, right? Okay. So now I've got 2 different phytoplankton and 3 different wavelengths. And so what do you notice? Where are they similar? Where might they give us a similar fluorescence? 470. And then at 440, population 1 is going to give you a lot more. And at 532, population 2 is going to give you a lot more. So this is the basis of how a multi-channel works. You have 3 wavelengths, but looking at the ratios of, say, this one to this one, or this one to this one, can give you information because each species is going to fill in sort of different amounts in here, okay? So that's how it works. And so you calibrate it the same way. You have all these cultures, you grow them to different, under different radiances, you collect them into growth phases, and you, for each culture, you do a dilution series, just like you guys did. But now you do it with a 3-channel fluorometer. And you measure its extracted chlorophyll, and you measure the fluorescence of the 3 channels, right? We're familiar? You calculate your slopes for your fluorometer, and so you do that for each culture. So my student, Chris Proctor, did this for a lot of cultures under a lot of different growth conditions. Each calibration, say this is culture, I don't know what this is, I made it up, grassless, okay. There's the slope for the green channel, that is this bar here. There's the slope for the blue channel, that's this bar here. It's the same species, it's just a different calibration slope. So what you see is that when you look across all of these different species, the calibration slope at each wavelength is different between its wavelengths and between the species. So what that says is if I look at the ratio of these calibration slopes, it's not telling me anything about concentration, but it's telling me about who's there. And so here's an example of what the 532 and 440 ratio looks like. So the Dinoflagellates have a very low 532 to 440. That's the ratio of this to this. The Synechococcus have a very high ratio. That's this to this. And you can see across the species that you can see statistical differences between these groups. So what? What would you do next?
Some seawater and attempt to get back at what species they are.
Exactly, right? So that's what you do. So we took it to the Arabian Sea. Anyone know what this is? So any text on this in here? 500 microns. Something giant. It's a Noctiluca. Noctiluca have been invading the Arabian Sea in the last 25 years. They were not really recorded before that. They're kind of taking over. They eat diatoms in addition to having pelagophyte Symbions. So this is what they look like in a beaker. So we did some transects. This was with Joaquin Goes and Helga Gomes, and this is the range of temperature and salinity along this transect. These are the fluorescence ratios from this instrument, and you see there's a lot of pattern. So we luckily had someone who looked under a microscope, and what we found was if we looked at how much Noctiluca was in the water by biomass, and we looked at the fluorescence ratio, when we had any sort of reasonable amount of Noctiluca, the fluorescence ratio was dominated by the fact that we had this one organism. So we picked stations where we only had diatoms, where we only had dinoflagellates, and we came up with characteristic ratios for each group. That's what these values are, the characteristic ratios. So then when we plotted the data, we could identify that this was a cluster of diatoms. This is the cluster of Noctiluca, and here's the cluster of Dinoflagellates at the surface. It turns out that those ratios move a little bit as you go deeper because they're putting on accessory pigments, which are changing their ratios as they acclimate to green light. But what it allowed us to do was to say that all of these points were mostly diatoms, these were Noctiluca, and these were Dinoflagellates. So we could take these measurements along our stations and convert it to who was most present. And it allowed us to see these Noctiluca populations. This is diatom. Station 7 is over here in the upwelling zone. It was all diatoms. That diatom population got subducted as you went offshore this way. And as we headed into these low chlorophyll waters, that's where all the Dinoflagellates were. And so there was a way of validating, we looked at microscopy to validate this, and it turns out that where we saw Noctiluca fluorescently, that's where we saw it under the scope. So it's a powerful tool for something that's so cheap. It's a cheap sensor, it's an eco sensor, it's a triplet like any other BBFL2 or BB2FL. And so it can provide you with a first guess of sort of the dominant type. But what it's telling you is something about pigments, something about absorption, and then you have to figure out what that means in terms of taxonomy. So it's not saving you anything, it's just giving you like a high resolution spatial pattern. So just one more tool in your toolkit. And there is one out on Globo. So if you want to look at that data, I'll show you how to get that data. So, okay. Oh, whatever. Okay. So pigments are relatively easy to measure if you're good in the lab. They are enticing products to work with because they seem to tell us something about something else that we want to know. They're a good proxy. They can be a proxy to a lot of different things, which should make you nervous. And so it's a proxy for a lot of things. I mean, it's not a good proxy for any one thing. But just take really a lot of care in education.
It's a huge group of people assemble both the different data one of the for models. So this is what is used mostly for models that are trying to different private items. This is what they use. That's sustainable and relatively large models.
But soon they're going to have IFCB data that will be available to them. And so that will be really interesting, but that's only providing you with a certain size range, right? And the nanoplankton, they're all little spheres in the IFCB. So it's going to have to be some combination of pigments and IFCB and other.
And these are standard format, relatively speaking, so it's easy for them to access them. And they have curated with different numbers that it's like, but all the pigments, you know, it's 90 pigments usually have the same material as in every HPLC. So it's easy to relatively speaking. If the IFCB, if you don't classify them in the same classes, some of them, it's not going to be as easy, so it's not going to be. So it's a real challenge to make the type of products that we produce as a community something that modelers can use to control from space or otherwise.
And it just takes time. I mean, I will say that when I was a graduate student, there was a lab that was just using HPLC, Carl Lorenzen's lab. And oh my God, this thing looked like someone, well, he probably did make it in his garage. I mean, it was like things everywhere, and like oscillators. That was just crazy. But his students were measuring pigments. And now 30 years later, that's the standard, and everyone can do it. You buy an HPLC, you follow the method, you get the output, calibrate it. But you know, yeah, now it's easy, but 30 years ago it wasn't. And so you can make, I mean, this is a point where you can contribute to coming up with data sets that are easy to read, easy to get to modelers, and meaningful, and have uncertainties around them. If this is a time, there's a huge opening for you to do this with optical data.
Another thing with HPLC, there was a crisis about 15 years ago, where it was discovered that the HPLC coming from certain lab did not compare well with quantum mass. And that was the lab that NASA sent out sample to that process. So these things are not, I mean, we say easy, but they're not easy.
And not easy. And that's what I mean here, right? It wasn't easy. And NASA had invested all of its money into one lab to provide pigments to the world, and that one lab got sloppy. So it's always good to have
Whatever method you use. there
validation.
is yeah, control, cross, so since then, a lot of labs are sending samples across the world to make sure that they're still doing things.
NASA asked for 10%. So if you're like, hey, I have an HPLC and I figured out how to do it yesterday, you know, I'm gonna send all my data to CBAS, they'll say, well, send us 10% of your samples and we'll compare that to make sure. And every time you do an experiment, send us 10% of your samples. And it keeps everybody on the same track. So. Yeah. Questions? What time is it now?
10:30.
Okay, so 10:45. Jeremy's gonna talk about getting size and taxonomy from reflectance. So now that you've seen some of this size taxonomy stuff with pigments and absorption, it's helpful to put that in context. All right, excellent. I don't know. Guillaume, did you stop recording? Guillaume, did you stop?